Okay, so good afternoon to everybody. We are going to start the session of the ESDR kitchen, and we are starting now the recipe book. And when we think about a recipe in general, we think about a set of instructions to make a dish, no, in general. But and it includes also a list of all the ingredients. So we are very curious to see the instructions and ingredients of Elisabetta Palazzo, and I pass the word to my colleague Enikö that will present Elisabetta.
Good afternoon, my name is Enikö Sonkoly and I work at Uppsala University in Sweden. Welcome to this episode of ESDR Kitchen. It is my great pleasure to introduce our speaker today. As Anna Balato said within the recipe book session. And our speaker is Elisabetta Palazzo, who is a researcher at the Dermolab Unimore at Modena, Italy. And Elisabetta Palazzo did her PhD in 2012 at the same place in molecular and regenerative medicine at the University of Modena, Italy, until under the tutoring of Professor Carlo Pincelli. After that, she also spent a few years at the NIH as a visiting fellow at the Laboratory of Skin Biology in Bethesda, Maryland, US. And after that, she came back to Italy and she's working mostly with skin cancer, animal models of skin cancer. And what is very exciting, she's working with zebrafish as a model for skin cancer. And this is what she's going to talk about today and reveal the secrets of how to work with zebrafish as a model for skin research. Elisabetta, the stage is yours.
Thank you so much. And thanks ESDR for the opportunity to be here today. So, okay. So today it's a really pleasure for me to show you the zebrafish model as a new and important tool for skin cancer research. So zebrafish, as you might know, is a freshwater fish. It is native of South Asia. Here you can see an adult organism, and sometimes it's sold under the trade name of aquarium of tropical fish. But in the recent year, it becomes to be considered a really powerful model organism in scientific research. Let's think about the field of neurobiology, cardiology, congenital birth defect, and cancer. And this is because of its unique features. And these features allow researchers to use it as a model organism for toxicological and pharmacological screening, for what is called phenotype-guided drug discovery, and for cancer study. So why using zebrafish? Why our lab, and we are not developmental biologists, becomes really interested in using this model? As I said, it has unique features. We can see that the embryo can develop outside the mother and they are transparent. And this can facilitate their observation using light cell imaging and their manipulation. So zebrafish can reach maturity and they are able to reproduce within 2 or 3 months, which is a relatively short time. And they can generate a lot of offspring. So a female can lay even 200 eggs a week, which means that after fertilization, we can use something like 300 larvae, and we can start our experiment with 25, 30 animals for each conditions. So we have a lower cost. We can we need less space. Just think about having 200 larvae into 4 petri dishes, but we will never fit 200 mice in the same space. So researchers began to understand how to use zebrafish for modeling human disease. And we can see that zebrafish genomes share the 70% of similarity with the human genome. And within this 70%, the 80% is composed of genes involved in human disease, so, like oncogenes or tumor suppressor genes. So we can have a specific we can follow specific pathways and we can target a specific biological process. So within the field of skin biology, we can see that this model has a different potentiality. We can study skin cell function, cell genetics, we can perform high throughput studies, skin modeling, skin disease modeling, and skin tumor modeling, which is the topic of today. So we are going to see a little bit in detail the development of the transgenic lines in order to obtain models of melanoma, and the xenografting model of cutaneous squamous cell carcinoma, which is the xenografting protocol is something that our lab developed and we use really frequently to obtain xenografting model with skin cancer. So melanoma, as you may know, presents heterogeneous mutation, but frequently arise from mutation in BRAF and RAS genes, which are implicated in the hematogenic pathway. So two zebrafish lines have been developed. The first one implies BRAF and p53 mutation. The other one implies RAS mutation. So regarding the first model, the zebrafish model of melanoma with BRAF mutation present BRAF mutated gene under the control of the MIRT promoter, which is a transcription factor involved in the melanocyte lineage. But in this condition, we can see that this zebrafish can develop ectopic nevi. But with the presence of p53 loss of function mutation, we can obtain the progression and obtain models of melanoma. The second model implies RAS mutation. So in this case, a researcher put RAS mutated gene under the control of the MIRT promoter. And in this case, they use also a reporter gene. So you can see here how it's possible to visualize melanoma, and the tumor can be developed in the same site as in the human disease. We have some limitation. For example, models with BRAF mutation presents long time to induce the tumor, and they may have different number of different step of mutation as compared to human melanoma. While zebrafish with RAS mutation show the rapid formation of melanoma, they need 4 weeks, and therefore they may not we will not be able to visualize the early stages. So this researcher performed other analysis and developed other beautiful model. So my goal today is to give you this ingredient as possible element for your recipe. And of course, choosing the right model will depend on the aim of your research. So let's go into the xenografting model. So xenografting could be performed using a cancer cell line or by using human primary cancer cells. So our aim is to model human cancer. So we are going to look at the invasion of cancer cells and the formation of different type of metastases. We're going to see immune response, and all the results we obtain by using cancer cell lines help us to obtain what people now call zebrafish avatar, which are patient-derived xenografting. Here you can see all the protocol that our lab use for obtaining a good labeling of skin cancer cells. And we're going to see this protocol in detail. Let me move this on this side. Okay, so we use two different kind of, we usually use two different kind of cells. That we can follow them really well. So in phase number 1, we can see we have to perform cell detachment, and we have to perform the staining in suspension, which we found is really the optimal condition. And then we need to perform the staining by placing our cell at 37 degrees for 30 minutes in basal media only. So see, these are the conditions for SCC, but we need to optimize the condition according to the specific cell line or the specific tumor we are working with. And then we need to resuspend our cells into a really small volume. Let me move again on the other side. In a very small volume. So at the end, we have a really high cell density suspension with something like 20-40 microliters. And then we are going to inject around 5 nanoliters into each larva. So here, the critical step will be to take into consideration the number of cells. In our experience, starting with 1 million cells, it's the optimal condition to guarantee that every time we inject a larva, we have the same number of cells, and so we have a really reproducible result. Of course, we need to optimize the labeling condition, as I said. Just for example, using SCC cell lines, we use 30 minutes, while if we use melanoma cells, we can use 20 minutes of staining protocol. And then we need to remember that not all cells are good for injection. And in our experience, using some SCC cell lines is not usable for, is not suitable for injection into zebrafish. So let's go into phase number 2, which is another critical step, because we need to, of course, perform the anesthesia of the animal, and then we need to align them really well into this plastic grid with methyl cellulose in order to have each animal in the same position and perform the injection with the capillary needle in a really reproducible way again. And this will, of course, avoid the damage also to the animal. And in this step, of course, we need to be really gentle every time we perform anesthesia, disposition, alignment of the zebrafish on the grid, and then we take them back into the petri dishes in order to guarantee a good and an optimal zebrafish survival. Then we go into phase number 3, which is a potential treatment we can perform on our zebrafish. So we can perform treatment either by injection into the yolk or by adding any drug, any compound you want to analyze into the fish water. So of course here it's obviously we recommend to perform a toxicity test in order to avoid damage again and reducing zebrafish survival and have optimal experimental condition. Then now zebrafish needs to be maintained at 33 degrees, which we found it is a sort of compromise between 28 degrees of the zebrafish and 37 degrees of human cells. And at this point, after a week, because we have a time point of a sort of time of 5 or 7 days post-injection, according to the presence or not of treatment. So we're going to analyze the presence of the percentage of zebrafish with a specific condition. So we may have in place condition where cells are in the same site of injection. We may have initial metastasis, which means having zebrafish with cells going outside the yolk, and have full metastasis, which means having zebrafish where cells are really going to be in a distant site from the initial site of injection. So now let me give you some example using some of our topic, which is the study of the neurotrophic receptor in cancer. And here we're going to see in cutaneous squamous cell carcinoma specifically. So CT71, for who is not familiar, is the common neurotrophic receptor. And we found that it has a function during epidermal differentiation, and it could act as a protective factor for SCC development. So So here we have a bunch of experiments we perform indicating that the expression of this receptor correlates with a reduction of proliferation, a reduction of invasion, and it can promote SCC cell differentiation. So we have this kind of phenotype when we perform CD71 overexpression, where we basically have a decrease of aggressiveness or invasiveness properties of SCC cells. So what happened when we use the zebrafish to understand the behavior of these cells? So here you can see very well what happened for CD71 overexpressing cells versus control cell. So you can see that we have a higher percentage of zebrafish having in place cells, while here you see a lot of cells going outside the yolk area. So we can count the percentage, the distribution of this type of metastasis formation, and you may also want to count the amount of cells or the red intensity that correspond to the cells into the animal. And we have really interesting results. So now we are going to see the developing of zebrafish avatar. So zebrafish users talk about zebrafish avatar to indicate the injection of patient-derived cancer cells. And it becomes a really nice model in the recent era because you can start with a lower number of cells. You can, of course, as I said at the beginning, you can reduce the cost, time, and you can overcome the heterogeneity with patient response to a specific drug or treatment. So let me give an example using our skin cancer model. So our lab, another topic of our lab is to the characterization of keratinocyte subpopulation in healthy and pathological condition. Here you have an example of keratinocyte subpopulation from SCC. So we were able to separate a population of more stem-like cells versus a population of a more differentiating cells. So we use these two types of cells to perform injection into the zebrafish. So we start from primary cells in this case. And this is what happened. So we were able to demonstrate, to find that the model is able to maintain the characteristics of the cells from patient. And you can see here that we have a higher number of metastases from what we call RAD, which are the rapid adhering cells, and collagen 4, which are the stem-like cells, versus the lower number of metastases we obtain with the non-RAD cells, which are the more differentiating cells. So we can perform this type of experiment, and we can perform expression functional study to obtain important information. And a thing that I will need to remember is that it's true that we are going to inject a really low number of cells, but for our experience, we need to start with a suspension of at least 500,000 cells, because the risk is to have a lower number of fish or zebrafish with exactly the same amount of cells. So the last thing I want to show you is the possibility to use zebrafish to evaluate the immune response. So here you see that we are in this area where the zebrafish larvae have the innate immunity. So we can measure the recruitment of macrophage or leukocyte within the site of injection. So we can analyze basically this area. So here is to show you what we can obtain by injecting SCC cells that you see in green, and using a model, a strategic model, where we can visualize macrophage or leukocyte. You can see here really well in red, macrophage or leukocyte recruitment, and in green, you see in real-time cell metastasis. To go back to our target, this is just another example. So here you see our cells in green going outside the yolk area in the control cells. And here you can see in with the red, the macrophage going into the site of injection. And then you can count the area, you can perform in situ hybridization. So we have several options to work with this type of model to measure the effect of on the immune system. So this is another clear example of the recruitment of the red cell that we can count, we can use the software, and so we can have a lot of answer using the same model with the cancer cell. So in conclusion, we have this beautiful tool that we can use for live cell imaging in vivo. We have similarity between human and zebrafish in terms of genetics and physiology of the innate immune system, which is really good for skin cancer. We can induce tumor in different ways. We can perform genome editing, and we can perform xenotransplantation, as I hope I show you today. And so zebrafish can be used for the study of different types of cancer, including skin cancer. So I would like to thank all people in the lab. You see this beautiful picture. And a special thank is to the Zebrafish facility and to Professor Natasha Tiso, which is responsible for the injection of our cells. And I would like to invite you to know more about the use of zebrafish in Tokyo. So hope to see you all there. Thank you so much. And I would like to take your question, of course.
Congratulations, Elisabetta. It's very, very interesting. And just to break the ice, I will start with a couple of questions. And I also invite all the people to make questions in the chat addressed to you. And my first question is actually a curiosity. How did you get interested in this model? So how did you start with zebrafish?
So, so we were looking for really a model that can give us all the answer we have about potential effect on our target on metastasis formation. We will need a model that could be really useful to investigate the effect of compound and the effect of genetic or ectopic manipulation of our target into cancer cell, which is something I mean, everybody's looking for, but zebrafish can give you the answer in a week, which is something really useful if you are going to, if you work with drug development, if you want to work with, if you want to have the answer, if your target is effective in producing a sort of answer for cancer cell. So that's why it's a really low cost, and people in the last 10 years did a really huge and effective experiment using zebrafish not only for skin cancer but for different type of cancer, starting from hematopoietic cancer to prostate or breast cancer. So it becomes really interesting for us, and the results we obtain with SCC, with melanoma were really interesting and convince a lot.
And my second question is actually is a really practical question, since we are in the recipe book. So you talked about primary tumor, no? So you need to collect, to digest from the primary tumor to collect cells. But yeah, how big is the sample you need for starting your model?
So sometimes we start with few millimeters.
Okay. So like a punch, I mean, like 3 millimeters is enough.
Yeah. So, but especially for SCC model. For other tumor, it will be a bit more complicated because you need to maintain and to expand the cells in culture in order to have at least, as I said, a sufficient number of cells to get the prep, the suspension that you want to inject into zebrafish. Because as I said, and the people in the review, that beautiful picture from the review I just showed, said, okay, you can use a really low number of cells for each larva, but you need to take into consideration that you need more cells to for the staining. You may lose some cells once you perform the injection, and again, after performing the injection, you can lose some larvae because not all of them are may not survive the procedure. So these are several things. There are several things that you need to take into consideration before start. So okay, for SEC, for example, which is mostly my field, we can start with a few millimeters. We can put them in culture. We can obtain a primary culture. So we have usually by co-culture with on a feeder, and then we can isolate our cells, and we obtain really a good number of cells, or bulk cells, to start for our preparation. So once we start, for example, with 1 million cells, if you start with 1 million cells, let's say with 1 million cells, we can inject really 100 larvae, 150 larvae. So if you consider that you want to have at least 25, 30 larvae for each condition, if you have a treatment, for example, if you want to start with this type of number, it's a good number from primary tumor, to start from the primary tumor. Okay. So
one last question by myself, then I will give space to everyone. And you mentioned about treatment, that in one of your slides you put that you test some drugs also.
We did. Yeah, we did. We tested. Sorry?
Which ones? Which drugs you tested in
so we are testing. We try to test some activator or inhibitor of our target. We were also testing some alkaloid compound. So we did several. We tested several different kind of substance or drug. Chemotherapy. We also tried some cisplatin, for example, to compare the effect of some activator or inhibitor as compared to a known drug for this type of cancer. But mostly by adding, so chemicals could be added to the fish water. But if you have, for example, some molecules, it will be nice to perform the injection. And this could be done the next day or the next couple of days after the injection of the cells.
Very nice models, indeed. We also have some questions from the audience, actually. So one of the questions is from Christine Pick-Bavastro, who is also thanking for your nice talk. So the question is it possible to perform xenografts with basal cell carcinoma cells?
Well, I would say, why not? We have never done with basal cell carcinoma, but I think that the method could be optimized with the for BCC. So I would say, yes, we perform also with primary keratinocytes after transduction. So and they were able also to create the tumor masses. So I think that also with BCC will be okay.
I guess the question was because BCC is so much dependent on the stroma, so you cannot really have cell lines kept in culture or cells kept in culture. But it's an interesting question indeed.
We could try. We could absolutely try.
Yeah, and then we have a question also from Cedric Preisenhausen asking if for live cell imaging of metastasis, do the larvae need to be immobilized? And if so, how would that interfere with circulation or other functions?
No, absolutely not. You can anesthetize the animal. And so for what we have seen there is no interference. We can immobilize the animal. Of course, this will not have, you don't need to do this for a long time, of course. But once you immobilize, you anesthetize the animal, you can immobilize the animal with methylcellulose, or we can use agarose, also low melting agarose in prepared using the fish water. You can perform the alignment of the animal on a slide, and you can perform the acquisition with confocal microscope. And you can actually see also it's really nice to see also circulation when you do acquisition with the microscope. And you can actually follow this. And I forgot to mention, but I know we had a lot of time, but you can also follow the same animal if you want, which means I can decide to follow a bunch of animal with that condition, and then I can decide to follow from day 1 to day 5. And this could give you more information about what's happening in that specific larvae or in a certain number of larvae.
Absolutely, that's really an advantage. One more question just from myself. Can you look at angiogenesis also, which is important for tumor formation?
Yes, there are some models. I think the transgenic animal we used once was the KDRL transgenic model. And so you can actually highlight the endothelial cells and using a reporter. And you can perform the injection as we did with macrophage reporter lines, and then see if you have an increase or any modification of the endothelial cells, because they can answer to the same way in the human to the presence of a tumor mass into the animal. So that's a possibility.
There's another question from the audience. It's from Neil Rajan, and he's asking for slow-growing cells. How long can the fish be kept alive? 4 days as a maximum?
So I would say no more than 7 days after the injection of the cells. So it's really important to, slow growing, but what are the properties of your cells? If you are maybe cells that can still have migratory capacity, maybe you can perform this experiment. Our cells are not slow growing, so you can see the difference sometimes when you have more differentiating cells or more differentiating cells as compared to others, but they are still migratory capacity, they are still mating capacity. So it depends mostly really on the properties on the cells.
I think we could talk about this for a very long time.
Yes, that's true. A lot of questions. And now that she was speaking about the difference, on the characteristics of the cells. Another question came to my mind, but I don't know. I don't know if I make it too many.
No, absolutely.
Maybe one more quick question.
Okay. When you, speaking about the squamous cell carcinoma, and taking into account that when you isolate cells from the primary tumor, do you take into account the grade of differentiation of the tumor?
Yeah, we experience some differences between well differentiated as compared to the moderately or poor differentiating cells. So this could be seen into the zebrafish model as well, because by using an MD SCC, we found actually there were a lot of metastases, while using a well-differentiated tumor to perform xenografting with primary cells, we have a lower number. Not that we don't have a metastasis, because they can form metastasis in zebrafish, but we have a little a lower number. So we are taking absolutely into account the grade of the tumor.
Okay.
Well, I'm afraid that time is up, but thank you very much for this wonderful presentation and all these fascinating images. Everything will be available at the ESDR's YouTube channel for those who missed the whole or part of the presentation. So thank you so much. And then we have some announcements to make. The next episode will actually also be a recipe book. So we will meet very soon, and that is the 19th of April, when Florian Gruber is going to talk about the epilipidome. So we will hear about lipidomics in the next recipe book episode, 19th of April. Yes, and the next announcement is about the ISID, which Elisabetta already mentioned, because she's going to be there. And for late breaking abstracts, the deadline is 31st of March. So I hope to see many of you in Tokyo. And then one more announcement, we have a lot today, is the State of the Art in Dermatology program, which is a webinar in support of Ukraine, organized by Christoph Schlabbach and colleagues from Ukraine. And the day one for this webinar will be on the 30th of March. Access is free for everyone. You can see more information on the ESDR website. And last but not least, the Future Leader Academy of Dermatology, organized by the ESDR, will take place in November 2023 in Madrid, Spain. So for those young colleagues who are interested in meeting other young colleagues and also mentors, this is a chance, and the application deadline is 14th of April. So thank you very much. And thank you once more, Elisabetta, for your nice presentation. Thank you, and wish you all a nice day. Bye.
Bye.
Bye.